welcome back to Devans Biology. In this video series of respiration in plants, we have completed the full mechanism of respiration, as well as in the last video we had a look on the total ATPs that were formed during the oxidation of one molecule of glucose. Now today let us see the enzymes that were used in glycolysis as well as their functions. You know, enzymes are very important in any mechanism. So we must know the enzymes used in the reactions and their functions. The first enzyme that is being used in glycolysis, in the first step of glycolysis, which was the first step when glucose gets converted into glucose 6-phosphate. The enzyme used was hexokinase. Now what is the function of a kinase enzyme? Kinase enzyme adds chemicals like phosphate to other molecules such as sugar or protein. So here in the first step where glucose was phosphorylated to glucose 6-phosphate, the enzyme which catalyzed this reaction was hexokinase. So whenever you see kinase word, then the function of that enzyme would be related to phosphoryl transfer. In the next step, when glucose 6-phosphate was converted into fructose 6-phosphate. Actually, it was isomerized into fructose 6-phosphate. Glucose 6-phosphate and fructose 6-phosphate are isomers. So, the enzyme that was being used was phosphoglucoisomerase. What is the function of isomerase enzyme? Obviously, isomerization means making isomer of the given compound. Isomerase enzyme catalyzes changes in one molecule. The end product has same molecular formula but different structure of the molecule. Now let us move to the third enzyme. In glycolysis, the third step was fructose 6-phosphate was converted into fructose 1,6-bisphosphate. Means one more phosphate was added in it. So we should say fructose 6-phosphate was phosphorylated to fructose 1,6-bisphosphate. And the enzyme which catalyzed this reaction was phosphofructokinase. What is the function of kinase enzyme? The function of kinase enzyme is phosphoryl transfer. Means it is adding the phosphate to a molecule. This enzyme is a very important enzyme. This is also known as, in short, we can say PFK. This enzyme is also known as pacemaker of glycolysis. You know what is the function of pacemaker? Pacemaker regulates the heartbeat. It can increase or decrease the heartbeat as per the requirement of the body. Similarly, PFK acts as a pacemaker for the glycolysis. Means it can increase or decrease the pace of the reaction according to the requirement of the cell. In the next reaction, when fructose 1,6-bisphosphate was cleaved into two molecules, the first one is PGAL, phosphoglyceraldehyde or glyceraldehyde 3-phosphate, and DHAP. Fructose 1,6-bisphosphate was a 6-carbon compound and these are 3-carbon compounds. The enzyme which catalyzed this reaction is aldolase enzyme. Fructose 1,6-bisphosphate is aldol. So it was cleaved or splitted into two molecules, PGAL and DHAP. So aldolase enzyme cleaves the aldol compound. This DHAP is dihydroxyacetone phosphate. PGAL and DHAP, these are isomers. So these are interconvertible. So this conversion is done by the help of enzyme phosphotriose isomerase. As I told you, these two are isomers. So which enzyme will catalyze the reaction? Isomerase enzyme. Isomerase enzyme, the function of isomerase enzyme is isomerization. What are isomers? Isomers are having same molecular formula but different structure, different physical structure, right? Now, the next reaction is in which PGAL, that is phosphoglyceraldehyde, gets converted into 1,3-bisphosphoglyceric acid. Converts into 1,3-bisphosphoglyceric acid. This reaction was catalyzed by glucose 3-phosphate dehydrogenase. In this reaction, we can see that one phosphate is also added. This was glyceraldehyde 3-phosphate, means one phosphate was present at third position. In this, we have two phosphates present at first and third position. So, in this reaction, dual function is being done. First one is phosphorylation, means addition of phosphate molecule, and second one is oxidation. So in this reaction, this enzyme catalyzes the function of phosphorylation coupled to oxidation. What is the function of dehydrogenase enzyme? Dehydrogenase enzyme removes hydrogen atoms instead of oxygen in oxidation reduction reactions. In the next reaction, in which 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid, the enzyme which catalyzes this reaction is phosphoglycerate transphosphorylase. Now, what is the function of transphosphorylase enzyme? It catalyzes transfer of phosphate group from one molecule to another. This reaction was substrate level phosphorylation. What is substrate level phosphorylation? Substrate level phosphorylation means synthesis of ATP directly from the metabolites. So here the phosphate group was transferred from one compound to the another. In the next reaction, where 3-phosphoglyceric acid was converted into 2-phosphoglyceric acid or 2-phosphoglycerate, the enzyme which catalyzes this reaction was phosphoglyceromutase. Now, what is the function of mutase enzyme? Mutase is an enzyme of isomerase class in which it catalyzes the movement of phosphate group from one position to another, not from one molecule to another. It just shifts the position. Here you can see in this 3-phosphoglyceric acid, the phosphate was at third position, and in this phosphate is at second position. The molecule is same, phosphoglyceric acid, but the position of phosphate is being changed. So this was done by the enzyme mutase. So in short, the function is phosphoryl shift. In the next reaction, in which 2-phosphoglyceric acid gets converted into phosphoenol pyruvate, the reaction was catalyzed by enolase enzyme. The function of enolase enzyme is dehydration. And the last reaction is phosphoenol pyruvate gets converted into pyruvic acid. You know, in the process of glycolysis, one molecule of glucose gets converted into 2 molecules of pyruvic acid. So, conversion of PEP, that is phosphoenol pyruvate, to pyruvic acid is the last reaction, and this reaction was catalyzed by enzyme pyruvate kinase. Kinase enzyme adds phosphate to another molecule. So in short, the function is phosphoryl transfer, that is transfer of phosphate molecule. Again, the last reaction was substrate level phosphorylation, and this enzyme is the action of this enzyme is inhibited by high levels of ATPs. So this leads to the negative feedback of pyruvate kinase. So these are the enzymes that catalyzed the reactions of glycolysis process, and these are the functions. The enzymes play a very important role in chemical reactions. So you must remember the enzymes and functions of each mechanism. Today we have completed the enzymes and their functions of glycolysis mechanism. In the next video, we will have a look on the enzymes, the location of enzyme and function of the enzymes of Krebs cycle. Glycolysis occurs in the cytosol of the cell, so all the enzymes were present in cytosol. If you have any doubt regarding this video, you can write in the comment box. Your doubts will be clarified immediately. Thank you.